The overall goal of the following experiment is to visualize the planes of ice bound by fluorescently labeled antifreeze proteins. This is achieved by growing single ice crystals to which the antifreeze proteins can bind. As a second step, each crystal is viewed through crossed polarizers to establish its singularity and orientation. Next, an ice crystal is mounted onto a temperature-controlled cold finger and formed into a hemisphere. It is then submerged into a solution of fluorescently labeled antifreeze proteins in order to absorb the antifreeze protein onto specific binding planes of the growing hemispherical ice. Results are obtained by visualizing and imaging ice bound by fluorescently labeled antifreeze protein using wavelength-specific light and filters in a darkened cold room.
The main advantage of this technique over existing methods like ice-etching is that the antifreeze proteins are fluorescently labeled to allow immediate visualization of the protein-bound ice planes.
Start crystal growing by preparing a temperature-controlled ethylene glycol bath at -0.5 degrees Celsius and finding a clean metal pan that fits into and can float on it. The crystals are formed in molds. Use cylindrical molds 3 to 4 centimeters high cut from a polyvinyl chloride pipe. Each mold should have a 1 millimeter wide, 2 millimeter high notch on one side. Apply a light film of vacuum grease to the side of the ring from which the notch was cut. Seal this greased, notched surface onto the metal pan with the notch oriented away from the center of the pan. Be sure not to fill or obstruct the notches with grease. Prepare as many molds